Hello everybody from the Netherlands, from Groningen. As you can see, we are proud from our tulips in the background. Good, so once more, yeah, thank you very much for having the opportunity to present ENPOD Nanotomy, short, large-scale electron microscopy database for type 1 diabetes. So today's topics will be first an introduction to my background and research interests. Then I will focus on nanotomy from the very start, 2011 till today. I will also touch upon color electron microscopy and then a special focus, of course, on ENPOD and type 1 diabetes. I will close with a final outlook for the future. So thank you indeed for introducing me, Kovai. This is my CV, my research profile, and I want to emphasize a few things. So it's nice to present at Pacific time. So here it's 8 o'clock in the evening. I did do my PhD at the Netherlands Cancer Institute. I was already in San Diego in the year 2000 with Matthias Volk. Then I moved to UCSD and did a postdoc with Mark Ellisman and Roger Chen. And actually only there I really became interested and I learned a lot on advanced microscopy, not only from Mark and Roger, but also from Tom Deering and Stephen Adams. So they asked me in Groningen, in the north of the Netherlands, to erect an advanced imaging center. And my special interest in this already for a decade is in type 1 diabetes. As most of you know, so we'll be very brief. The cause of type 1 diabetes is not known. The complications are really severe. The therapy is invasive and there's no cure. I'm also a cell biologist by training. So what I do admire in the human body or in the animal body is the beta cell. So if you compare the situation, if you don't have your beta cells anymore in type 1 diabetes, you have to measure your glucose, you have to measure that invasively, and also you have to provide insulin to your body invasively. So as a cell biologist, I'm always astonished again how nicely regulated the physiology is by this 1 gram of beta cells. And no, we human beings cannot yet mimic that. Back 150 years. If I teach and ask my students, do you know Gray's Anatomy? They start to laugh and everybody knows it. And then disclaimers, we never look at it. But actually, when I did my studies, medicine and biology kind of combined, we still had Gray's Anatomy actually to teach us the anatomy of the human body. And the atlas was actually presented in 1860. And this is how Gray then presented the pancreas. I named it because at that time, it was not known that you did have these kind of structures in the pancreas. It was only in 1869 that Paul Langerhans discovered these particular islets of cells together. So I think if I would ask you, what do you recognize, it still would be very difficult. But this would be the islet of Langerhans. Of course, and I will come back to that because that's sometimes lacking in electron microscopy, we need probes to be able to identify our targets. So this is actually an experiment which I performed in San Diego in collaboration with Vincenzo Cerulli. I will come back to him later today. But here I did label the nuclei of the cells blue, the insulin in green, and the glucagon in red. It's Friday night in the Netherlands, so it's actually time for a pub quiz, and I'm gonna ask you a few simple questions. And you can use the chat, but I don't see the chat yet myself. But if you feel free to use the chat, take on the challenge. I start very simple, don't be insulted, but the question is, are we looking at insulin or glucagon? So the girl on the right-hand side, insulin or glucagon? And as I mentioned, I cannot see this chat yet because I'm presenting, but feel free to share that amongst each other. I think this is a very easy question, so don't be insulted. If you are on a different level in type 1 diabetes research or seeing patients, the same question, insulin or glucagon, then this bottle. I think that's also still a very easy question. I typically use this also for the first year medical students. They might have a difficulty here. But then here, I would again ask it, insulin or glucagon, and then you might need some expertise or some previous knowledge. And as I explained earlier, I did label the glucagon with a red dye and the insulin with a green dye. But as most of you know, this is actually a human islet, but especially in the rat and the mouse islets, insulin is more central, and the alpha cells producing glucagon are more in the periphery of the islets. This might be another question, insulin or glucagon. So if you're more into chemistry or happen to know that insulin smells, it really stinks because of all the sulfur bridges. So this is insulin, the other peptide is glucagon. I think this might still be answered right by many of you. Typically, I would go to this slide then, insulin or glucagon, and then I actually hope that most of you are lost, because this is really key to my lecture today, how to recognize the different hormones and cells at the electron microscopy level or at the ultrastructural level. So I will actually not name which is insulin and glucagon, but we'll come back to that later. So my research interest definitely into type 1 diabetes. I also have another hat, being the scientific director of the UMCG Advanced Microscopy Center. So what we do with the electron microscopes regarding MPOT and nanotomy will be key in the remainder of my talk. So nanotomy, what is that exactly? And I was hesitant, but I decided, because most of us will be in our home offices at work, and we never see the inside of labs, especially not if you're in positions that you don't use the bench anymore. I'm excluded from the lab, but happily my team can just work and use the microscopes. So that's not too bad. But therefore you get a 3.5-minute tour in an electron microscopy lab, actually in my lab, and it also explains nanotomy. So, and you can also rehearse your Dutch.
Dit is Ben Giepmans, onderzoeker van de afdeling celbiologie, en hij doet onderzoek naar type 1 diabetes. Patiënten met type 1 diabetes moeten insuline injecteren omdat de insulineproducerende cellen in de alvleesklier om onduidelijke redenen vernietigd worden. Voor zijn onderzoek gebruikt hij onder andere elektronenmicroscopie, een techniek waarbij je wel tot 100.000 keer kunt inzoomen op biologisch weefsel. Om weefsels te kunnen bekijken, moeten ze eerst gefixeerd worden. Dat kan met chemische processen of met bijvoorbeeld bevriezing. Daarna kan het weefsel ingebed worden in hard materiaal. Vervolgens wordt het in hele dunne koeps gesneden, zo dun dat elektronen er doorheen kunnen. Als we 1 miljoen van deze dunne plakjes op elkaar leggen, is er ongeveer 5 centimeter. Dit is een van de allereerst gebruikte microscopen, gemaakt door Anthony van Leeuwenhoek. In de 20e eeuw is veel gebruik gemaakt van de elektronenmicroscoop die u hier ziet. In 2011 is deze uit rollatie genomen. De zeer dunne koeps worden in een vacuümkamer gesluist. De elektronen die van boven komen, kunnen dus alleen worden tegengehouden door het weefsel. Is er een grote dichtheid aan materiaal, dan kunnen we de elektronen aan de onderkant van het apparaat niet meer zien. Het beeld is zwart. Is er geen materiaal, dan worden de elektronen dus wel gedetecteerd, wat resulteert in een wit beeld. En grijs voor alles wat ertussenin zit. Maar een moderne elektronenmicroscoop is bijvoorbeeld deze, waar Ben Giepmans veel tijd opbrengt. Deze microscoop is geheel computergestuurd. Je kunt met een camera binnen in de microscoop kijken en daarna de beelden op het scherm zien verschijnen. Op de computer kun je dan tot in detail zien wat er onder de microscoop ligt. Het probleem van elektronenmicroscopie is dat de plaatjes zo gedetailleerd worden dat het lastig is de big picture te zien. Net als bij Google Earth, waar je op grote afstand je huis niet herkent, maar van dichtbij niet meer weet naar welke stad je kijkt. Daarom hebben wij samen met het LUMC een procedure ontwikkeld die we nanotomie noemen. Hiermee kun je losse beelden aan elkaar plakken, zodat je een heel groot en heel gedetailleerd beeld krijgt. Een foto van een paar miljard pixels dus. Deze techniek is nu nog uniek voor onderzoek, maar ook in het onderwijs voor medische studenten zullen wij het veelvuldig toepassen om het idee te krijgen hoe cellen in weefsels er echt uitzien. Heel anders dan in plaatjes in tekstboeken. Daarom hebben we de techniek hier voor u toepasbaar gemaakt in Eriba. U kunt zelf door een weefsel heen navigeren, net als een echte wetenschapper.
Yes, that's the anatomy procedure. I hope you enjoyed a peek again in the lab since a long time. I always do. I mentioned that we would use this a lot for education, and yes, we do this a lot. So all our medical students have a workshop on anatomy. This movie was like 10 years old approximately, so then we only had one data set. You saw the procedure in anatomy, but what we also did soon after is actually getting a website. And now I will go to this website, so I probably first stop sharing and then share again. So give me a minute to get my controls. So I think you now see the website, nanotomy.org. So here we have nanotomy to date. So as you can see, it's approximately 35 studies. The most recent one, not by us, but with other people, where we hosted data in the Lancet on COVID. Very popular, of course, these days. But let me show how this you can do yourself. I'm just now on the internet, how you can navigate through these data sets. And actually, I will choose this one. So now we see an islet of Langerhans within the exocrine pancreas. Yeah, so we really focused our imaging on the islets. This is an islet within the exocrine pancreas. So you can go wherever you like because the data is taken at high resolution, 2.5 nanometer pixel size, and you can just zoom in. And maybe you already recognize the nuclei, some blood vessels, empty capillary, a capillary with a red blood cell with an erythrocyte, a bunch of other cell types. And if we go to this area, this is my favorite area. You see different cell types actually. So you might see one cell type with very black granules, one cell type with more grayish granules, and one cell type with kind of crystalline granules. So I think this is quite convincingly showing that here we do have the glucagon alpha cells, glucagon producing cells. This is actually somatostatin, and you can recognize that by structure. And this is the insulin granules, which we also saw earlier. And these are easy to recognize because they're a little bit more crystalline. So insulin is a little bit more crystalline crystals compared to the others. But also this data set has a wealth of information. If you're looking at or interested in a fenestrae, you do see them here. An erythrocyte again, the caveolae. So wherever you go, you have a lot of information on this data set, and it's just accessible. So one thing in nanotomy is the data acquisition. The other thing is really to share the data. And if people want to address the ultrastructure of certain specimens, they just go to the website and can analyze these data sets. Now let me also zoom into a nucleus to show you that we could even detect nuclear pores if you have the right cross section. So that's a little bit challenging always. It's kind of unbiased to look for these kind of things. Oh, but here we have a nice nuclear pore. So I think this is the strength of nanotomy. This is the only example I will show right now, because I don't want to switch back and forth between PowerPoint and the web interface. But if we just simply go to the homepage, nanotomy.org, there's a few buttons here also for teaching. MPOT, I come back to that. Or actually I can show it. So MPOT, this will be shown later. People working in MPOT will recognize the numbers. You just click on a number and you get the electron microscopy of your donor. Yeah, so nanotomy, not only the acquisition, but definitely also the sharing of the data. Let me go back to my PowerPoint presentation. The data I showed you, like everything we do with new methods or advancing methods in microscopy, is on animals. So I briefly went to the MPOT data already, but most is on animals. And as I mentioned, we started this approximately 10 years ago. So when I was working on this in 2011, 2012, we were collaborating, as you heard in the movie, with the Leiden University Medical Center, with Raymond Ravelli, which you guys might not know. But in the same Leiden University Medical Center, Bart Roep was also working, and he definitely gave us advice. And we were focusing on islets, on a diabetic rat model. And he mentioned already MPOT, which was then still in the early stage, 2011, 2012, and that it might be worthwhile to team up with MPOT. In 2012, I did visit California again. Always very happy to visit after having been in the Scripps for half a year and in UCSD for over 4 years. Still am homesick now and then, but I was very happy to go to Monterey, California. And when I was presenting my poster, there was this lady coming up and she asked me about my rat data. Did you ever consider to use human islets of Langerhans? And then I mentioned to her that it was quite difficult for me to get hold on these islets of Langerhans. So this lady, Martha Campbell Thompson, then answered to me that it should not be a problem because she has a key role actually in the MPOT consortium. So MPOT, I think most of you are aware, so very brief, the network for pancreatic organ donors with diabetes, really focused on type 1 diabetes, collecting donor material, of course, not only type 1, but also control tissue. And in the past decade, also pre-diabetic, if you wish. So people positive for several antibodies, but no clinical signs of diabetes whatsoever. So that's the network, that's the samples they have, but the goal is really to understand how type 1 diabetes develops and even cure type 1 diabetes, like the JDRF goals. So they asked me actually to become the core, the large-scale electron microscopy core for MPOT. And what we did is getting their samples, so they're shipped to the Netherlands. We treat them such that we can perform the electron microscopy, the nanotomy. We put that on a special machine you saw in the movie, and then we acquire the data, make the stitches, and a stitch as this one I have been shown earlier today already. Yeah, so the MPOT, the network is present, really focused on the cause of type 1 diabetes and a cure for type 1 diabetes. The fixed islets, and this is still ongoing, so the database is still growing, are shipped to Groningen for nanotomy, and then we take it from a biobank into a data repository. Yeah, so the EM, the electron microscopy of donors, is easily accessible worldwide because it's only a mouse click away. So if people think EM is laborious, no, it's just a mouse click away whenever it's in the repository. So this is the NPOD repository. So I should admit, typically, if we don't publish, it's still under a password, or we don't share all the data yet, or only with collaborators. But once it's published, and this is a publication from last year with the NPOD group, everything is just open access available. And I will show an example later how people reuse that. So this is my introduction to large-scale electron microscopy or anatomy. So nanoanatomy, the anatomy of tissue, cells, organelles at a macromolecular level. Popular speaking, Google tissue. I think major benefits of nanotomy already have been shown. You have both the overview as well the high resolution. That also allows to image rare events. We see mitotic cells, we see centrioles, because we kind of image the whole specimen, really different from traditional electron microscopy when the operator select areas and just make snapshots of the image. So that makes it also very unbiased, both at the tissue level, but also up to the molecular level. And a big benefit is that we can open access, share the raw data. Nanotomy, as opposed to other things I will talk about, is really a routine technique for us these days. Another major benefit listed, but actually it's a huge disadvantage, is that we are looking at grayscale images, like Paul Langerhans. Yeah, so Paul Langerhans cleverly saw these chunks of cells together, but he of course did not have the immunoreagents that were developed later, let alone a fluorescence microscope. So to overcome this, I will take you actually to how we can combine other imaging modalities to identify what we're looking at. And then I will come back actually to the convincing images of the different cell types I showed earlier. Yeah, so if we would have labels that would label, for instance, the zymogen granules in green, so this is an immunolabeling overlaid on an electron microscopic image. So now in green, we can really clearly see where the zymogen granules are. And in yellowish, we can see where the insulin granules are. So you can really tell the difference of the cells. Probably way more convincing for you than what I first did on the electron microscopy image. But of course, these labels can also be used in an electron microscope. Something we explored already in San Diego, in the Ellisman lab, are the quantum dots that are both fluorescent, but they are also very electron dense. So these particles are quantum dots, and in this case, they are labeling the insulin. People in immuno-EM might be more familiar with immunogold labeling, so that's shown here. But in our hands, the quantum dot labeling is way more efficient. And of course, we have the added benefit that we also can see that in the light microscope. So there you can optimize your techniques. We made actually an ImageJ VG plugin, Gold Digger, immunogold quantum dots, so Gold Digger, that we can automatically recognize, in this case, the quantum dots. And you see most quantum dots are recognized, not all. So I think that's very nice, especially if you have these large images where if you would do this manually and you would have to annotate 1 million quantum dots, because that's what we see on these large images, you just cannot do that in your lifetime. So if we zoom out, these are collections of these little rounds, then again together in insulin granules. So these are all beta cells, and here you see that these cells indeed have distinct granules, but they're also not labeled with insulin. In this case, we used gold with a green circle to label a structure in heterochromatin, and again the other circles, the blue circles for insulin. What you can also see is that our Gold Digger sometimes also looks at the membrane, sees certain spots, and I don't know if these are quantum dots or if it's the membrane. Here I'm quite sure it would be the membrane here as well. So the Gold Digger is not really perfect. So the quest actually is for something that when we have labeling, we should not use Fiji algorithms, but we should just use another modality to define where our particles are, preferably just with an experimental approach. And that's what I want to present next, because I think that's very powerful. And there we are still sometimes struggling with the detection limit. So as I would say for nanotomy, just try it at home. It's a routine technique. This is still more tricky. Yeah, so a clear disclaimer. A sense of skills going from an islet of Langerhans, approximately 0.5 millimeter in diameter, to certain cells, tens of microns, the nucleus typically 10 micrometer, going to the granules that secrete hormones, typically like 200 nanometer. But we just zoom in more. Then we will see the insulin polypeptide, I would say approximately 1 nanometer in diameter, but let's zoom in a little bit more, and then we actually have the amino acid cysteine. Remember that I showed the cysteine bridges in insulin. That's very cysteine rich. Let's actually zoom in more on the sulfur atom. So now we are really at the atomic level, neutrons, protons, and the electrons in the shell, the sulfur atom. And these atoms, of course, we have in the electron microscope. We have in the incident electron beam. So this would be the electron beam in our electron microscope. And one thing which happens, and we typically do not record in an electron microscope, is when these incident electrons hit electrons of an atom and these electrons get dislodged, this is a very unstable situation. Yeah, so if from an inner shell an electron is dislodged, there's an energy unstable situation. Then an electron from an outer shell will fill this vacancy, but it has a surplus of energy. The surplus of energy is very typical for the atom, yeah, depending on how big the nucleus is. So this typical energy is released as an X-ray, yeah, but the X-ray energy is very typical for the atom. So if we would measure the energy of the X-ray, we would be able to define which atom we are. And I'm just looking typically at my mouse path. So this is my mouse path. So this typically takes like 15 seconds. But just by adding this additional detector, which we typically don't have in the biomedical sciences, at the medical centers, it's more for more basic research. But by just adding an X-ray detector to the microscope you saw in the movie, we can now also detect these X-rays. And then using my mouse path, depending on the energy of the X-rays, and I will zoom in a little bit, and the X-ray energy specific for the different atoms are mentioned here. I already showed you the quantum dot labeling, and the quantum dots have a cadmium shell. Yeah, so if you hit quantum dots, and there will be X-rays emitted, they will have these typical energies, 23 and 3. So if you look in red, in 23 there's a big peak, and there at 3 there's a smaller peak. Gold was also present in the sample I showed you, and gold has characteristic X-ray emitting of energies of 9.7 and 2.1. 2.1 small peak, and then 9.7 like here. So if we record the energy and then relate that to which atoms might release this energy, we can define which atoms are present. So if we do that in the picture I already saw, in which we submitted to Gold Digger to use algorithms to actually determine if this is more roundish and black gold than the other quantum dot particles, we now can just look at the cadmium signal. And indeed, from the insulin granules, you do see a big cadmium signal. And remember, insulin is very sulfur rich. So we also have a very strong sulfur signal in these granules. Yeah, so now we can actually extract more data from the electron microscope. We can also overlay that, and that's why this is popularly called color electron microscopy, or color EM for short. So we can really discriminate between the gold labels and the quantum dot labels in cadmium, and already the endogenous sulfur that I showed. Of course, we can look at other endogenous signals, like the phosphorus is really enriched in heterochromatin. So there we see a lot of signal, and that really co-localizes with where we did see the gold label for our heterochromatin structures. The phosphorus is also very abundant, of course, in mitochondria. That's why you see blue here at the screen. And the sky is the limit. I showed you my mouse plot, the periodic system, but you can actually address a lot of signals. So I will actually show a few very relevant signals in the islet. I hope you remember this islet. This is what I showed earlier, and that's actually why I try to navigate exactly to this area. And then I was actually quite convinced, but to be honest, I was convinced myself that I didn't convince you about the different cell types. I mentioned glucagon in black. So I was at a slightly other area, but this is then somatostatin, and this is then insulin because it's so crystalline. So I don't think I really convinced you. It would be hard to convince me. But if you go to the microscope and just look at the data, all endocrine granules are very rich in polypeptides, amino acids. So they all have a very rich nitrogen content, the amino acids. Insulin, we already saw, has a lot of sulfur bridges as opposed to the other hormones. So one cell apparently has more sulfur. And for one reason or the other, I don't know yet why, there seems to be more phosphorus in this cell. So we can, if we overlay this and make the nitrogen red, and in this case the osmium green and the sulfur blue, we can easily discriminate based on the atomic content the different cell types. So I think if I show you this image, you're convinced that these are the really different granules, different cells, and I already mentioned what kind of cells these are. So distinct granules based on elements. Also in our studies in the rat model of type 1 diabetes, we saw something very strange. So this is an electron micrograph of two parts of the cell at the border of the exocrine pancreas at the top and the endocrine pancreas at the bottom. This is the basal membrane in the middle. This is the nice endoplasmatic reticulum. These are the zymogen granules. I think you all recognize the mitochondria, another mitochondria and another one. But these are the zymogen granules. And these are the insulin crystals. Insulin crystals. This is glucagon again. And these zymogen granules really should not be here. They really should be in the exocrine pancreas. So we were really amazed. But then this is again interpretation based on the gray level scale on the ultrastructure. So is this really what's going on? So if we fingerprint this and use the color electron microscopy to see what's going on, indeed, we see the purple again because there's a lot of sulfur in the insulin granules. This is not labeled like that for the alpha cells, or sorry, the glucagon granules that should be in alpha cells. And of course, which really stands out are these zymogen granules, which have the same atomic fingerprint as the zymogen granules in these cells. Yeah, so here we do see that there's something going on, which we see more in the diabetic or the pre-diabetic rats than in the controls. So that's why we went back to the database and address if we also see these aberrant cells in MPOT nanotomy. So we have a lot of control donors, autoantibody positive donors, popular speaking pre-diabetic. Then we have the type 1 diabetic donors and also some type 2. And if we then address actually the different donors, and if there's the presence of these intermediate cells, we do see them in the control donors, but more abundant in the autoantibody positive donors and also in the type 1 diabetes donors. And if we were not sure whether or not it was really like zymogen granules, we again applied our color EM technique to define the fingerprint of the granules. Yeah, so also the color EM in MPOT shows us that we have these exocrine endocrine cells more in type 1 diabetes. Moreover, also the morphology is more affected in type 1 diabetes. That's why, as I mentioned, also in the control, we sometimes see these cells, but still a nice morphology with the mitochondria, a Golgi apparatus, for instance, more mitochondria, but really an affected morphology. This is insulin, it's outside vesicles, and this is a very stressed endoplasmatic reticulum. I can easily recognize that it's rough ER because I still see the ribosomes, but otherwise it really is aberrant. And that's what we always see in the autoantibody positive, triple autoantibody positive donors and type 1 diabetes. Yeah, so also with color EM, we see this. This is actually not completely surprising. We also see a phenotype of the exocrine pancreas, because I think another very nice and important outcome of the MPOT studies of Campbell Thompson and co-workers is that the pancreas is significantly reduced in type 1 diabetes and autoantibody positive donors, and also in first degree relatives. And not always, but there might be more causes for type 1 diabetes. But this really looks like if the effects on the exocrine pancreas precede type 1 diabetes. So it's not a consequence of a lack of insulin, but it might actually be causally related to developing type 1 diabetes. Yeah, so that's a major research question, which is still open. So large-scale EM, I already mentioned and focused this part on the grayscale data. That's not really a benefit, but that's why we have color EM, so we can combine either light and electron microscopy, or we can just look at the atomic fingerprints of our samples. Another benefit, which is really not a benefit, is a disadvantage, is that we are looking at fixed, non-living material. Yeah, so if we, for instance, in the exocrine pancreas want to know cause-consequence relationships, there's not much we can do with the fixed material of the donors. So back to the microscopy core. Actually, when I started 10 years ago, 15 years ago in Groningen, I was focusing more on advanced light microscopy first. These microscopes might look very impressive. Here we do have a couple of lasers and laser guidance boxes. So the lasers go into the microscope, but also a lot of equipment, actually, that's the biology in me, is just to maintain your samples at a proper level. Yeah, I already did live cell imaging in the previous century, and that always has been a struggle. And at a certain time, when all the microscopes were getting boxed, it was way easier actually to control the air. So this is actually just air control, 37 degrees, or depending on your specimen, maybe lower. It's humidified air, it's CO2 controlled, and also this box, also all this equipment, as you see above, is for climate control. So what we did a couple of years ago is addressing how cells in the brain of a zebrafish, so this is actually taken on that microscope, the microglia in this case, in the brain, we were addressing how these microglia interacted with neural damage. Yeah, so we can induce neural damage and then address how microglia is getting rid of this damage. So what we next want to do is something really similar. Now we have, again, the zebrafish model, but then the islets of Langerhans are labeled, or actually the beta cells are labeled with the green fluorescent proteins. So we can monitor these, and we can actually address what happens if we ablate or stress the exocrine pancreas. So mimic the situation, what we see in donors, something is going wrong, but now we have full control. We can do that with laser in the microscope and address what's going on. If you look at the ultrastructural level of the zebrafish pancreas, and this is after 5 days post-fertilization, so really in the larval stadium, we really see these zymogen granules looking alike zymogen granules I showed earlier, but also the endocrine pancreas really resembles what we see in mouse, rat, and human. So that's our next step there. We want to create exocrine malfunction either chemically or optically, and then see what the response is of the beta cells in the islet of Langerhans. So how would it look like in the microscope? First of all, I would show you that we will do this in a light sheet microscope. I don't know if some people on the call definitely will be aware or have one, use one. but I think this will revolutionize in vivo microscopy because it's so fast and it's less harmful than many other techniques we are currently using. So in a light sheet microscope, we can illuminate a sheet of the specimen and then record that everything which we actually illuminate. So every single photon we illuminate, we can record. And this is a very fast and sensitive system. So we can do that actually also with the zebrafish, maybe not only using GFP, the green fluorescent protein, but maybe GFP-based reporters. In this case, I show a calcium reporter, which is actually expressed in beta cells, and we can read out the oscillations of the beta cells. So the oscillations of the calcium over time is a direct measure of function of the beta cells. So this is what we do in collaboration with Elizabeth Carroll, and which we want to continue to further address if there is a cause-consequence relationship of what we see at the electron microscopy level, at the ultrastructural level. So that was just a little a step to in vivo imaging. I want to go back in the closing part of this webinar and the next steps in nanotomy. So one big disadvantage of our nanotomy is that we are scanning with a single beam, and we are scanning 2.5 nanometer pixels up to 1 square millimeter. So our microscope can easily be occupied for 4 days to make one map I showed you. So the question would be, can we engineer a better microscope? And then the answer is no, we cannot, or at least I cannot with my team. We are not engineers. I am collaborating with the University of Applied Sciences in Delft, and there was the idea to engineer a microscope not with a single beam, as I showed you, but with more beams, up to 64 beams. And that collaboration started, I think, approximately 5 years ago. There has been a spin-off company, and the spin-off company is called FastEM. For fast automated scanning transmission electron microscopy, and they launched already this system, and we did actually purchase one, and it will be delivered to the lab April 10th or something. This is taken from their brochure. I don't want to make too much advertisement about the microscope, but I really want to point you to the screen. Maybe you already recognize which samples they are using. So this is also from their brochure, where you see that they are scanning in parallel with 64 beams instead of 1. So this is, there's some other properties of the microscope that in the end, it's approximately 100 times faster than what we do now. Also from their brochure, scanning, in this case, an islet of Langerhans in the exocrine pancreas. This is a data set with our current microscope. It takes 75 minutes per gigapixel, and this is what the prototype FastEM already could do. And this will be further optimized, where we only take 4 minutes per pixel. So definitely 20 times faster, and that should resolve our bottleneck in acquisition. So islet microscopy, key signatures and niches. The nanotomy, please go to the website. Also, the multimodal imaging, I think, is very potent to identify what we are looking at. And we cannot always recognize it based on structure. So that's why we identify using other probes or even using endogenous atoms that can tell us what we're looking at. One of the things we actually saw in our rat study, but also in the ENPOD study, is that we have intermediate cells, exocrine, endocrine cell mixing, especially in pre-diabetic and diabetic donors. And I've stressed how important it is to have these probes for microscopy. So on the islet nanotomy, for this audience, what is next? So we launched this last year, the database. Bart Boep wrote a mini review about the insulin granules, and actually he went to the database and only picked actually something to illustrate his message. But that's definitely what we hope, that you use Nanotomy not only for illustration, but also get better insights in type 1 diabetes. And actually the latter, that's what Amelia Linneman did very recently. I think this has been published 31st of January this year, where she applied data mining from nanotomy.org as one of the techniques to study autophagy in type 1 diabetes, and that she found it was impaired in type 1 diabetes. Yeah, so we really hope that the data repository Endpot Nanotomy will be of use for you. We are not only happy to collaborate on Nanotomy, also on other things. So let me show you one example how we do that. When I decided from a very microscopy-oriented training with Mark Ellisman and Roger Chen, also try to find my own niche in biomedical sciences, where my original training was. In Mark's lab, I already collaborated with Vincenzo Cerulli. So actually the islets I showed you, the experiment was done by myself. But Vincenzo asked me to do this in a collaborative project. So the project was definitely his idea. But last Saturday night, he actually came back to me and saw the color EM, and he mentioned, I think that your element guided multicolor EM may be of tremendous help. So I replied to him, yeah, happy to collaborate, just send me your blogs. And I don't know what time it is in your time zone, but if you look at the clock, Friday, February 12th, so that's today, 11:49 a.m., I estimate that it's for most people 1 hour later. For me, it's a few more hours later. But the package has been delivered. So this is the blocks of Vincenzo. This is our ultra microtome. We will do the nanotomy. We will do some color EM, and also send him links back and together analyze the data. So closing, I think with the probes, anatomy, advanced microscopy in general, we have super duper resolution with this color EM using the X-ray detection. We can identify endogenous elements. We can paint our elements or identify labels, as I showed you. We can also quantify and identify probes if we do the correlated microscopy or with EM labels like Gold Digger. And we really have a high spatial temporal resolution up to the almost molecular level. And I think we are happily now in a revolution of combining the interdisciplinary research in computer sciences that also allow the data acquisition, but also importantly, the data sharing. But the next step we will take is going to a fair data sharing and try to team up nanotomy.org with international repositories. In the final slide, I would like to acknowledge my team. As I mentioned, all the data you saw, I'm never in the lab. I'm not allowed to be in the lab. But my team members are, so they generate the data, and we are still very active currently. I'm also very happy with how we get these microscopes. So the Dutch equivalent of NIH is NWO, has been very generous in applying us with equipment. And also more recently, actually, the medical center really made major investments. I do a lot on the advanced imaging methods with the team of Jacob Hogenboom at the Delft University of Technology. And I'm very happy to be part of the Endpot family. And especially I collaborate a lot with Irina, Marta, and Mark. And with that, I would also thank you, the participants of this webinar, for listening, and I hope you will be able to use the Endpot Nanotomy database. And also, if you have questions to collaborate, we really also want to share our equipment, and especially interested in applying that to islet biology to better understand the cause of type 1 diabetes and eventually prevent type 1 diabetes. Thank you very much.
Thank you, Ben. So I see there's some comment and a question under the chat. So I think first one is It is when you ask questions, there is an answer from Carmen. It's Inkshooting at the very first image. I think you show in the slide. And then can you see the comment, or I can okay. And then I think Anita said, great to see the Spark research is highlight. And then there is a UNPA also joined our ID initiative. And then there's one question is a zinc has been implicated in the insulin packaging in the granule. Do you see a zinc signal in your X-ray images?
Yeah, Ronald, I think it's indeed a very obvious question if you use the X-ray images to directly see if we have zinc signals. That's what we did as well. We don't see zinc. We don't see iron in the erythrocytes. We are now applying a method that people did not broadly apply in biology. What I think is that our optimization in electron microscopy, we take care of fixing our proteins first, our material, our proteins with aldehydes, preferably glutaraldehyde. We do that very carefully. So in animal studies, most people still infuse living animals with glutaraldehyde. We fix then our lipids with osmium. Yeah, so the lipids are fixed very nicely, but I'm afraid, coming to your question, that we lose a lot of ions, that we lose our zinc, that we also lose calcium. So also in the mitochondria, we have these crystals with calcium. We don't see the calcium. And as I mentioned, the iron and the erythrocytes, we also don't see. So I think that needs sample prep optimization, maybe high pressure freezing free substitution, or high pressure freezing and cryo-EM, but cryo-EM and EDX, where you need a high current beam, might not be compatible at this stage. So the short answer is no, we don't see it, but we are thinking about it. If you have other suggestions, Ronald, because we would like to see it, of course, they are most welcome.
Okay, and there is another comment and a question from Marianne. Nice to see you, Ben. Congratulations on the wonderful resource. I always associate you with Quantum Dot from our NICMR days. Would love to work with you on FAIR through DKNet. Are you going to expand to type 2 diabetes more, or is this focus going to remain in type 1 diabetes?
Yeah, so my focus is definitely in type 1 diabetes. So I don't have an intention to go to type 2 diabetes. Having said that, the other important line we are performing, and yeah, Marianne actually did work at NICMR at the same time as I did. If your new methods are successful, the most successful is if they're implemented in a lot of different research fields. So to that extent, I'm always open to collaborate with others, but my personal interest is in type 1 diabetes and islet biology.
Okay. If you have questions, you can also unmute yourself during this time or just add to the chat. Okay, there is another question from Carmen. I'm curious to know how deep into the tissue can the in vivo intravital image get? Could this technique be used to imaging diabetic mouse kidneys, for example?
Yeah, so we, Carmen, thank you for the question. We do have mice under the microscope, but these are typically window mice, and sometimes we're also imaging the liver. But the window mouse, of course, can go back into their cage and it can be imaged a few days later or a few weeks later. If we image the liver just on a cover slip, that will be a terminal experiment. So you could do something for that, like that, for the mice, but is that then intravital imaging? Because yeah, the animal is in vivo, but yeah, as I mentioned, it's a terminal experiment. We reach approximately up to 1 millimeter with 2 photon, and then really going to the infrared with an additional OPO added to the 2 photon laser. But that's kind of the limit, 1 millimeter.